ever wondered how scientists figure out which genes are active inside a cell? Today we'll learn about microarrays, a powerful tool which lets us see the upregulation and downregulation of thousands of genes simultaneously. A microarray is basically a chip containing thousands of DNA spots. Each spot represents a specific gene. When we run the experiment, we can see whether each gene is expressed more, less, or not at all. The process begins by taking mRNA from two samples. One is from a healthy cell and one is from a diseased cell. The mRNA from these two samples is converted into cDNA through reverse transcription. The cDNA is then labelled with fluorescent dyes. Green dye is used for the healthy cell, and red dye is used for the diseased cell. A microarray chip has many spots. Each spot is coated with multiple copies of a specific probe, which are short oligonucleotides that cover a sequence of a specific gene. When the fluorescently labelled cDNA from the two samples is mixed and added to the microarray chip, each strand hybridizes to its complementary probe on the gene spot. If only cDNA from the healthy sample is complementary to the probes on that spot, cDNA from the healthy sample will solely bind there. cDNA from the diseased sample remains unbound and is washed off. That spot on the microarray chip after laser scanning will glow green, indicating that the gene on that spot is only expressed in the healthy sample. If a different spot on the microarray chip contains probes that are only complementary to the cDNA from the diseased cell, only diseased cDNA will bind there. Healthy, unbound cDNA is washed off. That spot on the microarray chip will glow red, indicating that the gene on that spot is only expressed in the diseased sample. If cDNA from both healthy and diseased sample is complementary to the probes present on the spot, both of the cDNAs will bind. As a result, that spot on the microarray chip will appear yellow, indicating that the gene on that spot is expressed in both healthy and diseased sample. If none of the cDNAs from both healthy and diseased cells complementary to the probes, no binding occurs on that spot, and as a result, that gene on the spot is not expressed in any of the samples. That spot on the chip will remain grey. Applications. It is used in gene expression profiling, disease diagnosis and classification, genotyping, and SNP analysis. Advantages. It is a high-throughput analysis, cost-effective, and a relatively quick technique. Limitations. It requires prior knowledge of gene sequence, has limited dynamic range, and has lower sensitivity than sequencing methods.